Hello everyone. Welcome to one more chapter where we're going to discuss the nervous system infectious diseases. So this is chapter 22. So let's look, as always, to our learning objectives and define what we're going to see in each video. So this first video I'm going to talk about the definition of a central nervous system and meninges and all the structures and how they get affected. And then we're going to talk about bacterial meningitis. The second video, I'm going to talk about listeria, tetanus, and botulism and leprosy, which are other bacterial diseases. And then a third video with viral diseases of the nervous system, including polio and rabies. And the protozoan one that is relevant. And then in the last video, we can talk about the amoebic diseases and prion. Okay, so it's going to be 1, 2, 3, 4 videos. All right, so let's start by talking about the structure of the nervous system. So as you remember, so first, why is this important, right? These are the most devastating infectious diseases, the ones that affect the nervous system. And the consequences include deafness, blindness, learning disability, paralysis, and even death. So we're going to start by showing here to you that whatever is in yellow here is the nervous system. And the nervous system is divided into central nervous system, which is the brain and the spinal cord, and peripheral nervous system, which is all the peripheral nerves that exit from the spinal cord and the cranial nerves that exit from the brain. That's all peripheral nervous system. The meninges are those wrapping layers around the central nervous system, so around the brain and the spinal cord. And they're actually made by 3 layers. The pia mater, which is the one really attached to the nervous tissue. The arachnoid, which is this one in the middle, where we find the subarachnoid space. And the dura mater, which is this thicker one. The arachnoid is super important. So these 3 layers, before I say that, they wrap the brain and the whole spinal cord. Okay? Even the caudequina, this part, which is more loose, it's not like all in one dense structure as the spinal cord. Even the caudequina, which is the end of the spinal cord, is wrapped by the meninges. So what I was saying is that arachnoid matter is going to be a net with capillaries around. And in that subarachnoid space, you have drainage of the fluid that is called cerebrospinal fluid. So let's talk a little bit about that. The cerebrospinal fluid provides mechanical support, regulate ion composition, and maintain chemical stability in the brain. So it circulates in the ventricles, which are all these little blue areas that are here, and around. Same for spinal cord. It circulates through the central canal of the spinal cord and around the spinal cord. Okay, so if you have a trauma, for example, the fact that you have fluid around your nervous system acts as a shock absorbent, for example. So other than transporting all these ions from parts of the tissue to other parts of the tissue. What is important here? So as I said, so this is not the best definition for this picture, but I really like it because it shows you like all the features that we want to be looking at here. Subarachnoid space, which is this part here. What is important there? It's important that it filtrates, right? It drains the cerebrospinal fluid. So we start with the choroid plexus, which is here. And there are other ones here in other ventricles. And this choroid plexus is made of capillaries and epithelial cells. Remember the ependymal cells? that line the interior of these ventricles. So blood is pushed through the capillary and filters through here as cerebrospinal fluid. So cerebrospinal fluid is part of the blood, as we learn in A&P. And then that fluid is going to circulate here and then all around the brain and the spinal cord in the subarachnoid space. And then because this fluid is continuously being produced, it needs to be continuously drained. And the drainage happens mostly in the subarachnoid space, okay? And that is drained back into a sinus on the top of the skull, like under the skull, right on top of the brain. And that sinus is like dirty blood that is going to be drained back to the veins, okay? Another place where cerebrospinal fluid can be drained from is here. Remember the cribriform plate in your ethmoid bone? That plate, other than olfactory nerves, it also has these little capillaries that will receive this drainage from cerebrospinal fluid back to the blood, okay? So that's another place for lymphatic vessels that are present in the nasal mucosa. So there are two main barriers in the brain that are used as vital protection. One of them is the blood-brain barrier. So I'm gonna write here, since my slide doesn't have the best definition. Let's see if we can do this. So this is what we call blood-brain barrier, okay? And the blood-brain barrier is made of a blood vessel, or the capillaries that are all irrigating the nervous tissue inside the encephalon. And it's this blood vessel. So you're seeing a cross section of the blood vessel, right? You see how this is, the blood vessels here, these little capillaries, are hugged by astrocytes and by neurons. This hugging is a filter, is a barrier to avoid that substances that would be toxic for the brain pass through, okay? Another super important barrier is the subarachnoid space. In that case, we call it brain CSF barrier. Brain cerebrospinal fluid barrier. This barrier is also done by, whatever would go to the spinal fluid doesn't go because there is a blockage by the structures that are filtering the cerebrospinal fluid. So not everything from the blood comes to the cerebrospinal fluid. It's a way to protect it, right? You just pass good stuff to this fluid. But in the case of bacteria going through the blood and reaching these areas, it is easier to rupture this brain cerebrospinal fluid barrier than the blood-brain barrier. So these barriers are usually the ones broken when microbes want to invade the meninges and cause meningitis. I'm gonna write here, meningitis. Okay? So it's an important site of microbial entry. Now we're going to talk about the three types of brain infection: meningitis, inflammation of the meninges; encephalitis, inflammation of the brain; which we're going to talk a little bit less about. We're going to focus more on the meningitis, but that are vector-borne encephalitis that are super important and present risks. And meningoencephalitis, when it affects both meninges and the brain. And in this case, you can have, for example, listeria can cause meningoencephalitis. This is a good example. So we will start with bacterial diseases of the nervous system. So remember, there is always a triad of symptoms for you to detect. So what initially happens after that period of initial incubation? Initial symptoms are fever, headache, and stiff neck. Okay, the person is not really able to move the neck back and forth, to flex the neck or rotate the neck. Okay, and later on you would have other symptoms like nausea and vomiting. Right? These bacteria that cause meningitis also cause sepsis, so that's another complication that can come from bacterial meningitis. It can progress to convulsion and coma and death, because they can, then by sepsis cause shock and that general systemic inflammation. Viral meningitis, which we're going to talk very little, because they're more common, but they're more mild, so they're not going to be so tragic as the bacterial meningitis are. Okay. Okay, so there are three main bacteria that we're going to talk about: Haemophilus influenza, Neisseria meningitidis, and Streptococcus pneumoniae. The Haemophilus influenza is the one that has that antigen on the capsule called antigen B, and this is the antigen that is used for making vaccine against Haemophilus influenza. Important here, do not forget that this microbe, this bacterium, does not cause flu. It can cause otitis media, it can cause sepsis, it can cause pneumonia, but it cannot cause flu. Okay? So when they enter the bloodstream, what happens? That pathogen is going to have like some type of tropism to get to the meninges, right? And then they can break that blood cerebrospinal fluid barrier and affect the meninges. It mostly occurs in children 6 months to 4 years, and it is prevented by the Hib vaccine that is given as 4 doses at 2, 4, 6, and 12 months of age. Accounts for 45% of the bacterial meningitis, and it has a 6% mortality. And then we have Neisseria. This one, so the first one, the Haemophilus, and the last one I'm going to talk, the Streptococcus, are usually transmitted by through air, through coughing, sneezing, right, through droplets through the air. The Neisseria is more for contact to person to person. So here you need secretions from the throat or from breathing really close. So this is the one that is more common to transmit by kiss, by really close contact, which is very common in young people like teenagers, right? So in this case, individuals between 16 and 18-year-old are the ones more affected, and other than the vaccine that is used against the serogroups A, C, Y, W, and B, W, not the B. There is another vaccine that's called MenB that includes the antigen B, which is very relevant for teenagers. So it's common to see outbreaks in dorms and military barracks, where people were very close together and there is more physical contact for reasons you know. And mortality here with antibiotic therapy is between 9 and 12%, without 80%. Okay? symptoms that are common in the meningococcal meningitis, the one caused by Neisseria, are fever, vomiting, headache, neck stiffness, light sensitivity, and lethargy. You also have loss of appetite, diarrhea, irritability, and crying when it's the case of infants, right? And one thing that I'm gonna mention to you later is that if we go back here, you're gonna see that here you have a gram-negative Diplococcus, right? Here for the Haemophilus, you have a gram-negative rod. So one is a rod, gram-negative. The other is a Diplococcus, gram-negative. And then you have Streptococcus, which is a coccus, gram-positive. Remember, name ends in coccus, gram-positive. So this is also encapsulated, so they're all encapsulated, but it's a gram-positive. The Streptococcal pneumoniae meningitis is the one called pneumococcal meningitis. It also, the Streptococcus pneumoniae can cause pneumonia and otitis media, and it's very common in children, ages between 1 month and 4 years. So again, these bacteria, the same as the Haemophilus influenza, are transmitted by coughing, sneezing, while people are in close contact with others and they're breathing the bacteria, right? Remember that another option for meningitis could be E. coli in kids, okay? And they can get it by eating food that is contaminated, and that could cause meningitis in children because they still don't have a perfectly active and mature immune system. This is showing what happens in the meninges when an infection, when a Streptococcus pneumoniae infection happens, is taking place. Look, all this whitish part is pus. It's a purulent type of infection, right? So it recruits leukocytes to the area, and the meninges has a lot of pus. That's why we're going to talk about the spinal tap. When they remove the spinal fluid, it's common to see that fluid is cloudy, turbid, right? So a little bit about diagnosis. If you have a suspicion of meningitis due to that triad of symptoms, you usually do a spinal tap. So it's a lumbar puncture, which is characterized by injecting a needle close to in the caudequina roots, but not in it, around it, to remove the fluid from the subarachnoid space. So this, you see, happens between L4 and L5, the lumbar vertebrae. There are other ways to, like if you do antibody agglutination test, it's a way to detect. But a first test that you do here is to see if there are pathogens in the cerebrospinal fluid. And you do that by performing a gram stain. So with the gram stain, this is what you see. For the 4 bacteria that we talked about, the 3 plus Listeria that we're going to talk in a minute that can cause meningitis or meningoencephalitis, they look different when it comes to the gram color and when it comes to their shape. So combining morphology with gram stain, you can get really close to the actual diagnosis. That will of course be confirmed by other tests. But it's so simple because you just spread the bacteria from the cerebrospinal fluid on a slide, you fix it, you do a gram, and you're really close to the final diagnosis. So if it is Streptococcus pneumoniae, you know that they are Diplococcus, purple, gram-positive, right? So this is what is showing here. If you have Listeria as the bacterium causing the meningitis, then remember they're gram-positive and they're bacillus. So they're not going to look at like 2 little cocci together. It's going to be one long rod, okay? And remember that strep means chain, so that's why they look like a little chain here for the Streptococcus. And then now the 2 gram-negative, the Neisseria, which is also encapsulated, and you can see very clearly that they're Diplococcus, and the Haemophilus influenza, which is a broad and it's pink, also gram-negative. So they're either gram-negative or gram-positive, and then the combination of being Diplococcus here and here, and Bacillus in the Listeria and Haemophilus. So with this is a very good tool for a differential diagnosis. So when you look at the cerebrospinal fluid, other than the gram stain and the turbidity, they usually count mononuclear cells, which are usually higher than normal. Sorry, less glucose in the case of bacterium and normal glucose in the case of virus. So there is low glucose because the bacteria are using that glucose to survive and grow. So you see a lower ratio of glucose in that fluid. You have more protein than normal, and also elevated pressure because you start to produce more fluid because that fluid is not being functional, right? That's all I wanted to say. It's less turbid in the case of viral meningitis, and there are some differences that can be addressed. If there is no pathogen, for example, when you're looking at the slide and you don't see any bacterium, then you have a suspicion of virus meningitis. Then you need to use other tests to detect it. So Listeria, since we're talking about the meningitis, this is going to be our last one, is caused by Listeria monocytogenes. That's a gram-positive rod, like I just said. I love this picture because this picture shows you how they can spread from macrophage to macrophage, and they induce membrane ruffling, right? So they induce, so you see, this is one, let's see if I can draw here. This is one macrophage that is emitting a pseudopod. See, that's one, right? And that pseudopod is has a bacterium here in the middle. And then this other macrophage is also trying to phagocyte. Oops. That's the other macrophage here, right? Trying to phagocyte that pseudopod from macrophage number 1. And that's how the bacteria kind of induces all this by inducing membrane ruffling and projection of the cytoskeleton of these macrophages, and then they jump from one macrophage to the next. Usually this disease is foodborne and asymptomatic. You don't have a lot of very serious meningitis cases in adults, but they are very common in children, okay? Around 1,600 people get listeriosis each year, and about 260 people die in the United States. This infection is more likely to seek in pregnant women and the newborns, so not individuals that are not pregnant or age between teenage and 65 years old, but also older people over 65 have a bigger tendency because of the weak immune system. It is very common to see Listeria contaminating deli meats, and in that case it's important to avoid it by not storing deli meat for longer than 2 weeks and in the proper temperature, right? For pregnant women, that's why they say avoid deli meat, avoid spreads like meat spreads, meat pate, because they usually could, be contaminated and usually cause a disease in someone that is pregnant. If a store package is open with deli meat or anything that contains processed meat, do not eat or leave in the fridge for longer than 5 days. This is from the CDC, guys. This is super serious. And you say, well, but if it's in the fridge, it's going to be fine. No, it's not, because they grow in the fridge. Listeria is a psychotroph. So they grow in the fridge. The disease that is going to cause, like I said, is mild in adults, but worse in people that are immunocompromised and kids. The risks for the newborn are abortion, and they can even be stillborn. Okay, and can cause a lot of neurological issues. Okay, so next video we're going to talk about tetanus, forward 2 or 3 more infectious diseases, and then we move on to the virus. I will see you in a minute.